So I'm very pleased today to have Ajit Kaushik. Is that the right pronunciation?
Yes, it is.
Good. And you're the assistant professor of chemistry at Florida Polytechnic. Is that right?
Yes, it's
Yeah.
correct.
And a PhD is it in chemistry?
Yes.
Okay. So, and also widely published. Well, I can let you say you have very many awards and accolades in the Indian Society of Chemistry, I think. Is that right?
Yes,
Yeah.
I'm fellow there.
Okay. And the reason we set up this call is about a paper that you recently wrote, Manipulative Magnetic Nanomedicine, and that raises, a lot of questions for ordinary people. What is this about? Because this year we've had to all interest ourselves in coronavirus and vaccines, and people are still confused a little bit about what this science really is. And I wonder if it's possible to explain in a kind of accessible way, what this miniaturization of medicines really means.
Yes, this is really nice to connect with you, Alex. Thank you so much. And you're right, I am a chemist, and beside teaching chemistry to the engineers, I am working in the field of developing miniaturized biosensing prototype. And at the same time, I am developing nano-systems which can carry a drug to a particular location in the body for maximum therapeutics. So you are right, we are in a tough situation, especially I would say coronavirus pandemic. And the question was how we can control this. Second, how we can treat this. Third, how we can manage this. So these three questions are in the mind of everybody, and this predictive solutions. First, we have to detect it. Second, we have to detect it selectively, no confusion. Mm-hmm. Third, we have to treat it precisely. Mm-hmm. So I would like to touch on these aspects one by one. First, how we can detect it. So detection means we need something which can detect the presence of this virus very selectively. So everybody or every health agency in the world was in rush to decide what to do. This virus was new. We were not having any tool which can detect this virus accurately because there was no information available. So that time, experts, virologists, and computational biologists or expert of infectious diseases, they sat together and gave some ideas. Let's do this, let's do this, let's do this. This is too much into the detail. So their first focus was to find some biomarkers which are very much selective to the actual virus. And these biomarkers were used first to detect this virus for diagnostic point of view. And these biomarkers were adapted by biotech companies to make a rapid testing kit. And this testing kit was adopted by all the agencies so that they can do rapid testing. Another challenge was that this virus is different. This is not like the virus came like Zika, Ebola, where we control the mosquito population and disease went away. Mm-hmm. This is something that transmits from human to human. We cannot compromise human to stop virus. So we need different tools. So in that case, if I got coronavirus, not only the accurate detection I need, I also need the tracking of myself, where I'm going, where I'm sitting, to whom I am meeting with, I'm attending some function or not, because I may carry virus and transmit millions of people in a longer time. So that time, computational approaches emerged to make models where we can do tracking of infection by putting something in cell phone. That's why these different apps came into the picture. And these apps were very, very useful. So we have a rapid testing system. We have an app. Now we were able to understand how this virus is spreading. Different predictive models came into the picture. So now the second stage, we were able to detect the virus. Can we use this detection for understanding viral infection? Or can we connect this information to know which therapy is good and which therapy is not? So in that case, we need to have a very selective detection of marker. That time, nanotechnology appears. People were thinking about making biosensors for quantitative and selective detection of this virus at a very low concentration. So these kind of sensors were useful to monitor the infection because these sensors perform rapidly. So we can do tests many times in a day on an individual. So these sensors are very small, easy to carry. Any clinic, any health worker, even patient can test themselves. That's why if I can operate something to know how I am feeling, it means I'm taking care of myself. Or a technology can be tuned to perform for everybody. That's why this is a personalized approach. Means one device, one person. Or I'm a patient, I need a device, I can test myself. So personalized approaches were the demand. And how we can make it personalized when detection of virus is easy. Second, the bioinformatics detection means I'm getting something in numbers, and that numbers is a kind of information, and that information can be known as bioinformation. Then we do bioinformatics analytics to understand what is happening more. So that detection information can be shared with medical center immediately if we add the component of Internet of Medical Things. This is a new technique which are connecting with sensing prototypes and sending patient information directly to the medical center. So the medical center can analyze that data and can tell to the health worker, hey, you send me the information, this is the viral load and this is serious, so give this medicine to the patient. So I'm connecting the dots and everything comes like a telemedicine. Mm-hmm. So we can perform testing wherever is the patient. We can sharing that information somewhere else in a medical center and experts sitting in the medical center telling best treatment to the health worker who is perform testing. Mm-hmm. So if I am living in a remote area, and someone is performing testing on me, it's so impressive. I don't need to go to clinic. I don't need expensive equipment. I don't need high-class equipped laboratory where my sample should go and then next day I receive the information. So making it fast, biosensors were in demand to manage this coronavirus infection. So this is all about how to detect a virus or how we can monitor the progress of this virus, whether someone is under the treatment or someone is not under the treatment. The sensor means this is giving bioinformatics about a patient. And the patient-to-patient things changes. That's why this is known as personalized disease management approach. Mm-hmm. And biosensing is a class of science that deals with sensors that has bio component. Bio means biomolecule. They can be enzyme, antibodies, DNA, RNA, aptamers. So when we develop a sensing prototype using this biomolecule, we call them biosensors. And overall, biosensors understand any response, a biological response, and that device converts that response to a number. And that number is always in correlation with the concentration of a targeted marker like coronavirus. Suppose I make a prototype which is specific to coronavirus, then I will be having a chip. Biosensor can be a chip. Chip means tiny miniaturized stuff. If this chip is getting a response when I put the real sample, real coronavirus sample on top of the chip, then through the display, I will get a number. This number will tell me this is the viral load of a patient. So that viral load, that information will be analyzed by an expert and say, hey, this is significant, or this is non-significant, or this is too serious. Based on that, healthcare can be managed. Like someone has to go to a hospital, or someone can be cured at home. So there's a lot of angle of biosensors in health management.
And just to understand, is it you have to put something? Is the biosensor in you, or is it you put like blood or spit or whatever on something else to get the reading? How does that?
This is true. So biosensing is a substrate. It's a kind of platform. We have to put real sample on top of that.
Yeah. Okay.
So that can be blood.
Yeah.
Saliva.
Yeah.
And urine.
Yeah.
So when we talk about a large population testing, we try to avoid blood because blood is infectious.
Yeah.
And we try to avoid urine because when we are thinking about a viral infection, means it's in a large number of people. So we cannot rely on collection of urine and testing. In that case, saliva is always a preferred biomarker or real sample to perform sensing. And so now things come how we can make sensors very effective. First thing, when we are dealing with virus, a biosensor must detect virus at very low concentration. So here is the science, technology. So we have to develop this biosensing prototype on nanomaterials. Because these nanomaterials are very small nanostructures. Nano means you can say that 1 billion nano equal to 1 meter. And nano cannot be seen by our eyes. It's like million times smaller than the hair, our hair. So why do we work on nanomaterials? Because this is the size of a material where material is most active, or material exhibit its maximum best performance. It can be optical performance or electrical performance. So when we are dealing with very small concentration of a virus, then this small concentration will produce a very small response. And that small response should be detected by advanced electronics. It's a combination of material science, biomedical engineering, and electrical engineering, so that we can sense this very small response, which is very important, using smart electronics.
And I just wondering, I mean, part of that is, the old way of having a case was for someone to be ill, and then, someone suspects they have something, and then you go off and get that tested and say, okay, you are ill with this disease. But now, I mean, I think one of the questions now is, people are being told they're ill because maybe it is a very low concentration, or so they're asymptomatic now. Is this new?
Yes.
Is a new term? I mean, what do you think about that in terms of someone really being ill? If you say to someone, we've done the biosensor, you've got a very low concentration, and that makes you then a case. Does that actually tell you much
Yes.
about how they feel or how infectious they are?
Yes. So let me try to clarify this a little bit. Now, symptomatic carriers or asymptomatic carriers. Symptomatic means I got coronavirus and I have every symptoms. Means I'm very sick.
Mm-hmm.
Asymptomatic means I am sick, I have virus, but I'm not sick.
Mm-hmm.
So I have no symptom. So why should I go to hospital?
Mm-hmm.
So let me refer my previous statement. That's why tracking of a patient is very important.
Mm-hmm.
So whoever has virus, whether symptomatic or asymptomatic, they get it from somewhere.
Mm-hmm.
If I'm an asymptomatic, means I have virus, but I'm not showing symptoms. But
Mm-hmm.
if I know where I'm coming from, then I can understand, oh, I met with a guy who has virus. Let me check myself.
Mm-hmm. Mm-hmm.
That kind of awareness we need.
Mm-hmm.
So now the significance of low virus is we can do early stage disease diagnostics. If I knew that I have started getting sick, it's easy to manage my
Mm-hmm.
infection.
Mm-hmm. Mm-hmm.
If the virus go to replicate in the body in very large amount, then someone has to go to hospital.
Mm-hmm. Mm-hmm.
So another application of nano biosensor is early disease diagnostics.
Mm-hmm.
Whether you are asymptomatic or you are started getting sick, it works equally for everyone.
Mm-hmm. Mm-hmm.
Yes.
But presumably the treatment would be totally different then. I mean, this is the big debate that
Yeah.
there's
So,
been.
yes, this is one side of my research, biosensor.
Yeah.
Second side of my research is nanomedicine,
Yeah.
developing nanomedicine.
Yeah.
This is more like treatment side. So I don't investigate new drugs. This is
Yeah.
not my expertise.
Yeah.
But if you see that some drugs are there,
Yeah.
and they are effective drug, but the challenge is that this drug has to go to right place.
Yeah. Yeah.
And sometimes the place we are targeting has a lot of barriers.
Yeah.
Our body is a complex system. It works perfect, but it's a complex system. So if I need to send a drug to a right place, now I have two approach. Either I put a lot of drug in an individual that some part goes to the right place and some goes here and there, I don't care.
Yeah.
This approach is a normal practice.
Yeah.
And that's why most of the patient got side effect. Their side effects are sometimes because of the diseases and sometimes because of the excessive drug we have in the body. The drug has to do something in the body. If the drug is going to the healthy organs, of course, this is going to affect.
Yeah.
That's why at early stage, most of the drug predicted against coronavirus has a lot of side effect.
Mm-hmm.
They are affecting eyesight, they're affecting heart functions, people are feeling fatigue, people are feeling back pain. So these are the side effects.
Yeah.
So the question is that how can I use the small drug and maximum benefits without side effects, without any bad effect?
Mm-hmm.
So my idea is that what if I load the drug on nanoparticles, and then these nanoparticles will go to the place where I want to have the drug, and then we release the drug.
Yeah.
And these nanoparticles will consume in the body without causing any side effect, and the patient will get right amount of medicine at right place.
Yeah.
So this is called nanomedicine approach.
Yeah.
And this also has the concept of pharmacology And this also has the concept of drug delivery system. So, and at the same time, coronavirus has uh, multiple serious concern, but two concerns are very important. First, I have to remove this virus from my body. Second, we all know that this virus compromises our immune system. What if I give something to a patient that improve or boost up the immune system of an individual? So I was advocating two drugs at the same time. First, which can eradicate the virus from the body, and second, which can support the immune system of infected people. So this was like a multi-drug approach, and the delivery of that drug using a nanoparticle. I said magnetic nanomedicine because my previous research was based on developing nanomedicine for the treatment of HIV/AIDS in the brain. I observed, or I have demonstrated this, that if I have a nanomedicine, which is magnetic in nature, then I can control the delivery of this cargo by an external magnet. And we were able to send drug from our bloodstream to the brain by the alignment of some magnetic forces. So I projected this in my article that this drug has to go to liver, this drug has to go to brain, because there's a lot of correlation between coronavirus and neurological disorder. And this drug has to go to the gut, which is also a very protected part of the body, and most of the drug does not go there. So my idea is that if we use a magnetic nanomedicine that has two drugs, one is antivirus, second one is immunoboosting approach, and we can tailor the delivery of this drug either in liver, brain, or gut, maybe 3 positions at once. Then we can control this virus completely, and patient will recover very fast. So that was my idea about that paper.
But people obviously will panic with the very idea that you have a magnetic field that can move the drug into different directions, and there'll be questions about how do you avoid mistakes with the magnetic field.
I respect their concern. Their concern sometimes gave us the new direction to work. And maybe I need to work more on awareness. And the point is that whatever we demonstrate in our lab, always tested on recommended doses. We used recommended dose. We do proper calculations, and before going to the animal model, we test everything in vitro level. So first we test everything on brain cells, then we optimize our number, and then we translate this number according to the targeted testing species. Same number will not work for in vitro, same number will not work for mice model, same number will not work for monkey, and same number will not work for humans. It's like a step by step. It's a lot of steps. And every time we have to be careful. We have to follow all the guidelines given by health agencies. And we have to submit various reports, and expert review our reports. Then we go to the next level. Yes, this is a new field, no doubt. And people concerns are very important for us, so that we can correct every process according to the best healthcare.
But if just doing on the carrier part now, if I summarize correctly, what you're saying is that you can actually cut the dosage down in theory quite a lot, but you're actually delivering to the right place. Therefore, you need much less. In this case, it would be the mRNA, would be a much lower dosage because it's going to where it needs to go to, right?
Yes, like there's another angle of nanomedicine is the protection.
Mm-hmm.
The microRNA are very tiny biomolecules,
Yeah.
and they are not stable.
Yeah.
If I put in the body, my body has a lot of complex biomolecule. They can eat these microRNA immediately.
Yeah.
And the same concept, this microRNA will not go to the right place.
Yeah.
So even the present vaccine is an example of nanomedicine because
Yeah.
they are protecting this vaccine by a biopolymer, which is either lipid or PEG.
Yeah.
And it's working.
But it's working, but you're saying it's because it's not, you can't sort of direct it, it's still a big dose in order to get.
Yes.
Okay.
Yes. So maybe in future we have more better carrier that those will be magnetically controlled to reduce the amount of injection, and in the less amount we can have better effect.
But in terms of, it gets more complicated, but in terms of, you're saying the simplicity of what you were doing, that sounds like it would be more complicated because you would need to get the right magnetic field and you'd need to direct the nanoparticles.
It might be very difficult to visualize, but it's not. It's a simple injection.
Yeah.
You just relax maybe 30-40 minutes and drug will be at right place.
But I mean, how will it go if you need to sort of steer it as such? How will it get to the right place?
Okay, so when we design a nanomedicine, first we have to localize the drug, which is in a broader uniformity, and then we always functionalize our nanomedicine with reference to the organ. So if I'm talking about liver cells or brain cell or gut cells, they always have very specific biomarkers. So there's a lot of various layer by layer functionalization in nanomedicine, so that drug goes to a very targeted site. It goes there, it sit over there, and then drug release in a sequence, because we have to avoid drug to drug interaction. If I am putting two drugs, these two drugs can combine with each other and there will be no effect. So it's a systematic formation of this nanomedicine. At every step, we have to characterize whether I'm getting it or not. A lot of it's a tidy process to make it, but once it's formed, it's very simple. Simply an injection, like maybe 30-40 minutes, relax under a magnetic field, and that magnetic field is very, very smaller than the magnetic field we have in an MRI machine. If I'm using like 3 Tesla MRI, that may have side effect, though I'm not aware. But the magnetic field we used to demonstrate this in a mice was 0.8 Tesla, not even 1 Tesla. And in 1 hour, drug was inside the brain. So I have to work a lot to make it more simple so that people understand this. And I'm sure one day magnetic nanomedicine will be revolutionary.
And then in the meantime, how do you people will say that this medicine then is tied up with mobile telephony and things like that. How do you say no? This is just to do with a very light magnetic field, and we know it works, and it's got nothing to do. These other theories are wrong, basically.
No. That's why I was saying that we have to work on awareness side also. If I'm putting magnetic field, I have to tell this is not something like MRI or no, it's very tiny magnet that we have in our transistor, like a ring, and you can put it around, no problem.
Okay,
it's a very simple approach.
Yeah, that bit's clear. Just wondering, going back to the testing, I'm still trying to understand how it's more, let's say, you're saying, if you're a long way from somewhere in a rural area, I'm just trying to understand how the technology of putting in the sort of nano sensors, having the device that transmits to the data center and the
Yes.
Internet of Things, that sounds quite complicated compared to just doing have a spit in a cup and send it
Right.
off for a
Yeah.
PCR test.
This is a very good question. So I think it has multiple steps. So first step, I will be making a nanoparticle based sensing chip.
Mm-hmm.
So that sensing chip will be a sensing prototype. And now the technology is very nice. This sensing chip directly goes inside the cell phone, and I can do testing using the cell phone.
Mm-hmm.
So this tiny chip is very, very small. We can easily hold it. And then we have some manifold which has a sample area where we have to drop the real sample.
Mm-hmm.
So first I have a sensing chip, then I drop the real sample, and the
Mm-hmm.
real sample means saliva. It's
Mm-hmm.
kind of simple spit. Within 30 minutes, my sensor will sense the virus in the saliva.
Mm-hmm.
And then this reading will go to the cell phone.
Mm-hmm.
Because now the chip is connected with the cell phone,
Mm-hmm.
and the real saliva is on top of the chip. Within 30 minutes, I will get reading in the cell phone.
Mm-hmm.
So now I have an algorithm over there that if this is my response, then how much will be the virus load?
Mm-hmm.
This will be an immediate conversion.
Mm-hmm. Mm-hmm.
So when I know that this is the viral load of a patient in a rural area,
Mm-hmm.
now every cell phone has a lot of data sharing options.
Mm-hmm.
We have cloud, we have OneNote, we have Microsoft system.
Mm-hmm.
That data sharing is very easy nowadays.
Mm-hmm.
So that connection from cell phone to a medical center will be done by Internet of Medical Things.
Mm-hmm.
And once data is there, everyone can have the access of that and analyze the informations. What I'm saying analysis, because it's not only the value of the virus is that information is going to the medical center. I can, if I'm testing it myself, I can put some number. This is my report. I am feeling high blood pressure because nowadays everyone has a smartwatch.
Mm-hmm.
I'm having high blood pressure. My blood sugar is low. I'm feeling fatigue and I'm not feeling hungry.
Mm-hmm.
So someone who is sitting in medical center, like suppose you are a doctor, you will read the note. Okay, a value came. Person Ajit, having viral load this, who is not feeling hungry. Blood pressure is down. Sugar level is down. Then if you are a doctor, you can decide, or you can pass this note to your superior, like, hey, this is my opinion. What do you think?
Mm-hmm. Mm-hmm.
And then I get the message back. Ajit, we analyze your data. You have to take this medicine and send us your report tomorrow again.
Mm-hmm. Mm-hmm.
And this is happening.
Mm-hmm. Mm-hmm. Mm-hmm.
So that's why sensing prototype, cell phone, and data sharing is all connected in a row.
Yeah, yeah. But I think what you're saying makes sense in a lot of ways, that you understand the viral load. I think a frustration of what's been going on now is that we just get these positive tests. It's just come back positive, and you get these thousands of cases of quite a lot of young people. And a lot of those tests, they've sort of rerun them, and they haven't been done in accordance with the manufacturer's instructions. There's been a lot of false positives. I mean, it feels like in this case we've generated, we just say, okay, that's a positive, this is bad. We report the number, and then that gets us into lockdown, and it seems to be a bit of a cycle, too much dependent on these PCR test numbers. But what you're saying is a differentiation between a low virus load and a high virus load, but that's not what we're seeing right now, is that? Is that fair with the PCR testing?
This is true. We have to work more on data analysis.
Mm-hmm.
Reporting a number is not solving our purpose. We are not managing the diseases. We are informing people that you are sick or not.
Mm-hmm. Mm-hmm.
So if we can analyze that informatics with reference to the medical condition of a patient, then we can use it more nicely.
Yeah, yeah.
That's why people are saying it like intelligent healthcare.
Yeah, yeah. Well, I think it's very interesting. Hopefully other people also find this interesting. It's nice to hear from someone who is doing good or on a mission to do good. And because I think there can be a lot of fear around the kind of science that you're talking about. And well, hopefully, this explanation will help to put some of that fear to rest.
Great, great. And thank you for having me. I really shared my thoughts very openly.
Yeah.
And if you have any concern, I'm here.
Sure. I'll put a link also to the paper so people can read for themselves.
Yes. Thank you. Thank you,
Okay.
Ali. I wish
All
you
right.
a great.
Okay. Thank you very much, Ajit. Thank you. Bye.
Bye.
Bye.